focus on viral phylogenetics and epidemiology. His research typically focuses on HIV, but during the SARS-CoV-19 pandemic, he has shifted his focus to studying SARS-CoV-2. Professor Niema is not only known for his technical skills, but he's also very popular among the students at UCSD, so we are very excited to have him speak at SC Hacks. Without further ado, please welcome Professor Niema.
Thanks so much. Thanks for having me. I'm going to just whiteboard it, if that's okay. I feel like it's a little bit easier. And yeah, if people have any questions throughout, like feel free to interject. I'll have the chat open if you want to type any questions in the chat. But yeah, cool. So basically in the world of bioinformatics, I'm hoping people have taken like maybe an intro bio class or something like that, but the term central dogma should hopefully sound familiar to people, or if you haven't taken a bio class, that's okay. This will be your TLDR of it. But basically this is the central concept of molecular biology, where you have DNA, which then gets transcribed into RNA. And then you have RNA gets translated into protein. And protein is kind of the functional unit of a cell. It's like the core, kind of the smallest unit of doing something that a cell can have. And proteins can like work together in complexes, whatever you want to do. And this central dogma is basically existent in pretty much any organism. So whether it be humans, whether it be bacteria, or even viruses, this kind of forward direction flow of information is pretty standard. So how does this relate to SARS-CoV-2? So SARS-CoV-2, it's actually an RNA virus. So we have this like RNA virus, it's single-stranded, and it has some genome sequence, which we can think of as roughly 29,000 letter string over the alphabet A, C, G, U. And when the virus first broke out, one of the kind of interesting problems was, what is the genome sequence of this unknown virus? And that was kind of what researchers were focused on back last December. But since then, now we know for the most part what is the DNA sequence of this. So we have what's called a reference genome. We have a high quality, RNA sequence. We have a high quality genomic sequence of the SARS-CoV-2 virus that we can now do future comparison studies against. So we have some basis for this. Okay, so what's the practicality of this? In practice, there's some very interesting public health related questions that people want to be able to answer. So some questions might be like, what are the variants? I'll say like what are the variants of COVID-19? Like how are they spreading? Things like, are transmissions happening within my city? I don't know, there's a bunch of different questions we can ask. And kind of the most basic question is, does person X have COVID-19? But yeah, so there's some kind of larger scale questions that we can ask at a population level, and then there's kind of smaller level questions that we can ask at like the individual level. And in this talk, I'm going to give a very brief overview of how we can use bioinformatics to try to answer these types of public health related questions. So first, I'm going to go on the kind of basic level, and then I'm going to branch out to more general. So first, this question of like, does person X have COVID-19? And even more specifically, like what variant of COVID-19 does this person have? So to answer this, what people usually do is like, let's say that we have this person over here. I'm going to draw them as a sad face because they're not very happy. They're feeling sick. We do I'm not a very good artist, so bear with me. You do the nasal swab, and then now we can get basically, this looks like a carrot, but this is a test tube. We get a sample of the coronavirus or whatever is going on in this person's sinus, which if they have COVID-19, we get a sample of the SARS-CoV-2 virus. And then once we have our sample, we can run it through a big sequencing machine. So I'm going to put Illumina, maybe trademark. I don't have the I don't have the rights to this or whatever. Right. So Illumina, it's a company down the street from UCSD. And if you get a bunch of samples from people, you can kind of put these samples onto one of these sequencing machines, and via some magic that is way out of the scope of this very short talk, out comes a bunch of short sequences. So here we have sequences. So these are like actual like A, C, G, T, whatever sequences. Each of them is roughly length somewhere between like 100 to 150. And the reason for this is because the sequencing technologies, they're not able to just kind of read this genome from start to end, letter by letter. Instead, what they're able to do is read random short fragments of this genome. The sequencing machine has no idea what order it's reading these fragments from. It doesn't know like where in the original genome they were. It just can read a bunch of tiny fragments. So us as bioinformaticians, or as like the tech people, we're given this data set, a bunch of sequences from some given person. And now we want to try to figure out what variants do they have and like what does their actual genomic sequence look like. So what we do is we take that reference genome. I'm going to call it ref. This is the reference genome that is that high confidence genome that is made available to us. And we take each of these short strings. So maybe I'll color code them. So maybe this first one was actually like a red one. Maybe this next one was orange. Let me actually use my eraser. So let's say like the first one was red. The second one was orange. The third one was green. And maybe like the fourth one was blue. So we can use tools that basically are going to try to see where does this exist in the reference genome. So I have the sequence and I'm looking for where does this sequence appear as a substring, or at least a substring plus or minus a few changes somewhere in the reference genome. And let's say this one happened to match to this part of the reference genome, and this part happened to match to this part. So we basically are taking each of these sequences and seeing where in this kind of high confidence genome they best match up. So this is called read mapping. And there's a few different tools that do this. One tool is called Minimap2. One tool is called BWA-MEM. And the various tools, they have various levels of multi-threading. They have like various levels of optimization and accuracy. But yeah, these are kind of two common tools that are used for read mapping. Okay, and then once I know where each of these short sequences existed in this long sequence, what we do is basically, so maybe I'm going to draw a couple more of these. So these are also some short sequences that just kind of match up randomly. The next thing that we do is called pileup. So we basically try to see for each position of the reference genome, what are all the different letters that the reads from our sample had at that position. So maybe in the reference genome, this is an A, versus in the different reads that I obtained, these short sequences, maybe this one is a C, this one's a C, this one's a C. So that tells us that at this position, this person actually has a C, even though the true, like the reference virus had an A in it. And then like maybe at this position, the reference is a C, and then maybe each of these says C. So here the person matches with the reference versus here the person was different. But basically we do this pileup to get an idea of what are the most frequent letters that we see at every single position of the reference genome. And then using this, so we get a pileup. And then to do this, people use a tool called SAMtools. And then from the pileup, we can do what's called variant calling, and also consensus. So variant calling is basically just getting a list of any places in the genome that our sample differed from the reference. So for example, it would say at this position here, the reference had an A, but our sequence had a C. And then maybe at this position, the reference had a T, our sequence had an A. So anywhere that our sequence seems to differ from the reference, variant calling is just listing out all those positions with like what was the position, what was the letter in the reference, what was the letter in our sample. And then consensus is now assembling those variants, kind of like plugging them in and actually getting a single long sequence that is unique to our sample. So our sample's genome. Kind of in one step, we can do these two things. And the standard tool that's been used for this is called IVAR. I'm just going to maybe circle the tools. Okay, so at this point, we now basically, given one person, have a genome sequenced, like can put together from that person. So we can now answer questions like, does this person have COVID-19? I could have seen, was I able to get a sequence? Was I able to actually match any of these to the COVID-19 genome, or did they not match? I can answer questions like what variant is this strain that what strain does this person have? I can look at this variant calling output and see, based on these mutations, where in the kind of global data set does this person's viral sequence look like it might fit? So yeah, so this is for a single individual. And then now if we do this process for a bunch of different individuals, so let's say from person 1, I got some genome sequence. From person 2, I got some genome sequence. From person 3, I got a genome sequence. From person 4, I got a genome sequence. And like from a bunch of people, I get a sequence of their genome using this exact workflow for each person. Now we want to start asking some kind of more global level questions. So going back to these questions, so these two kind of went hand in hand. These were for the individual versus these questions. Now I want to answer for my population. So I've sequenced a bunch of people in my population. I want to see how are the variants spreading? Maybe what is the distribution of the variants? And are the transmissions happening within my city, or are they kind of people getting infected from outside and coming into my city? So to answer this, what we ultimately want to do is estimate the evolutionary history of the virus. So what we can do is first, given these genomes, these are the genome sequences that we obtained like this, we can perform a task called multiple sequence alignment that basically just lines them up. So like, let's say this sequence was like A, C, G, T, A, C, T, and then maybe this sequence was like A, C, G, T, A, C, G, T. By an alignment, what we mean is like these two sequences don't seem to match up very well. But if I just stick some spaces in between different parts of it, so if I put a space here and I put a space here, so let's draw how that would look like. So if I put kind of like spaces, so A, C, G, T, A, C dash T versus here, if I put A, and then I put a space in between the A and the C, and then I kept copying. Now, by just adding these two spaces, these sequences line up very nicely. So the first step is given a bunch of sequences. I basically line them up so that the matching letters from each sequence are all in the same column. And then given my multiple sequence alignment, I can now do what's called phylogenetic inference. Oh, and I forgot to list tools. So some tools here. One tool that people use is called MAFT. And then here, some tool. All right, so yeah, this is MAFT. And then also, I'm going to say, these are some tools that people use. And then, yeah, so given the multiple sequence alignment, we now do what's called phylogenetic inference. A phylogeny is just an evolutionary tree. So we're basically given this using tools to try to reconstruct the evolutionary history. So maybe this is like 1, 2, 3, 4, and 5. So like maybe this is person 1 and person 2 are the most similar. And then maybe like person 4 and 5 are also very similar. And then maybe 3 is closest related to 4 and 5. And then we have like some ancestral state. So some standard tools that people use for this are like IQ, tree 2, or fast tree 2. Okay, and then given this evolutionary history, we can then overlay any demographic information that we have about these people. So for example, if I know when these people got infected, I can overlay that information and try to scale the evolutionary history to be kind of telling the story over time rather than just looking at the relationships between sequences. If I knew what country these people came from, or like what city or some geographical information, I could overlay that as well. So for example, if I saw that these people were from like the USA, maybe these person were from Europe, and then this person was from like Australia, I don't know. This would show that the virus kind of spread to Australia and then Europe and separately spread to the USA. So like if you overlay the geographic information, you can actually use the evolutionary history to try to trace how the virus is spreading across the world or across your community or whatever you're interested in. Okay, so I know I'm kind of throwing a lot at everyone and it's probably like extremely rapid, but I just want to give you a broad overview. And then during SD hacks, if this kind of realm of questions is interesting to you, you can explore each of these a little bit closer. So now kind of for the important question, what is there that at a hackathon you'd be able to work on? So if I can generate this is extremely helpful to public health officials. So as a computational person, I want to try to generate an evolutionary tree as accurately as possible and as quickly as possible. Ideally, I want to be able to update this every day so that in real time, public health officials can make these types of important decisions on like, do we want to restrict travel? Do we want to close restaurants? Like what is going to actually have the biggest impact? So in this workflow, so kind of the individual level workflow, the kind of slow step is generally this beginning part, the read mapping. So Minimap2 and BWAMM, they're two of the more common tools that are being used to do this. Minimap2 is the fastest tool right now, but still, it doesn't scale as nicely as we would want it to scale. So like read mapping is really the computational bottleneck of this workflow. So maybe I'll like circle it in green. So if you were to somehow accelerate this step, either implementing on GPU, because most of these tools are implemented on CPU, or if you implement on FPGA, or if you came up with some clever algorithm to maybe skip some computations that you don't need, that would be a very nice step to accelerate. In this workflow, viral MSA is pretty quick, but this step, the phylogenetic inference, is extremely slow. So in this entire kind of COVID-19 world, this single computational step is by far the slowest. So on a data set of even like maybe, I want to say like 100,000 people, this step can take upwards of like 24 hours of compute at least. So we're looking at like days of compute time to do this on like even reasonably small data sets. Yeah, so if you wanted to look for projects that you want to like speed things up, those would be the two most important steps. If you wanted to look for projects to kind of like package this analysis in a nice workflow, so bioinformaticians like me know how to run these types of tools from the command line. But the people that are really doing this like sample collection and like doing the brunt of the work, they're mainly biologists that don't necessarily have as they might not be as knowledgeable on the computational side of things. They might not know how to use high performance computing clusters. They might not be comfortable with the Unix command line, whereas most of these tools are Unix. So if you wanted to do some type of like web app development, having a nice packaged web app or some type of user-friendly workflow that handles running this workflow or this workflow, either cloud-based or on the user's computer, but in some way that has like a user-friendly setup, that would also be a very interesting potential project. Yeah, and then if you wanted to come up with like custom hardware solutions, kind of each of these different stages would benefit from acceleration from custom hardware. Yeah, so that's kind of, and okay, and then the last thing that's really interesting would be, so just running these analyses is, it's interesting, but the real interesting part is what can you do with the output? So like given an evolutionary tree, how can I visualize it? How can I make sense of it? So I guess the last potential project area that I would recommend if you wanted to make impact here is some way of visualizing and like in a user-friendly manner, interacting with these types of data sets. So just real quick, I'm gonna maybe share my screen. I'm gonna pull up a tool that might give people a little bit of inspiration on that front. There's this tool called NextStrain that maybe I'll end with this. So NextStrain basically throughout the entire COVID-19 pandemic has been maintaining global data sets of like evolutionary trees. So here, like, let's say I want to look at a global level one versus I can zoom in on a specific continent, but for now global. Each leaf in this tree is one sample. So like this leaf here is a sample from the USA. I can't really see when it was collected on February 13th. And we have some information about some of like the interesting mutations that it has. And they're all overlain with their lineage information here, but I can also change this to like maybe country. Maybe I want to overlay country information. So like these ones, the red ones are all from, does it show? Is there a key somewhere? So these ones are from the USA. These ones are from Australia. Yeah, cool stuff. So some way of like in a user-friendly way, someone being able to kind of click on something and interact with it and get helpful information from it would also be interesting. Cool. Okay, so I think there's like 4 minutes left, so I'll kind of stop there and leave room for if people have any questions or if anyone's interested in potentially doing a project in this space, if you want some pointers or anything like that. Maybe I'll end it on. I'll just leave a slide with some of the tools. Oops. But yeah, so feel free to like ask any questions if anyone has any. Let me get my chat open. I think people should be able to unmute if they want, but I don't know. So there we go. So tools. So for multiple sequence alignment. Actually, maybe I'll just type it. That might be faster. Text. Alignment. We have viral USA, math, inference. We have tree 2. We have fast tree 2. For read mapping, we have map 2 and that's probably the important ones. Cool. Any questions? Any? I don't know. No questions?
What could be some approach to making read mapping on a GPU? Like, what tools could be could come in handy there? Or not tools, I mean, we're building a tool, but like what knowledge, you know?
Yeah, that's a good question. So for Minimap 2, I guess in 2 minutes, I'll try to do a very quick summary of what Minimap 2 does. But basically, a lot of these tools, what they try to do is, so pairwise. So I'll give you some pointers to read at, like some pointers that you can Google to like read a little bit more on. Pairwise alignment, there's an algorithm called Smith-Waterman, like Water MAN algorithm. So this algorithm, if you're given a string of length k and a string of length n, it can align two sequences. So if this was like the reference genome, this was one of our reads, it does it in big O of n times k. So what a lot of tools do, including these tools, is they try to take my read of length k, and then they try to like fragment it into smaller pieces that they know for sure one of these is guaranteed to match perfectly somewhere in my genome. And then they use hash-based techniques to try to find those perfect matches. So you pre-process your reference genome, keeping track of where certain substrings exist in some form of a lookup table. And then you do some similar processing on your read to find where short substrings would show up in the reference genome based on that lookup table. And then you can kind of do localized alignment operations around those perfect matches and then kind of string them together. So I would highly recommend looking at the Minimap2 paper. But in general, the kind of overarching thing that would help is any way that you can do very fast lookup table stuff. So any type of hashing that you can optimize would probably speed up Minimap2 nicely. Cool. I think I'm out of time, but if anyone wants to ask any follow-up questions, or as you're working on stuff, if you want any like resources that I can point you to, feel free to email me and I'm happy to help out. Cool.
Thank you so
Thanks
much to the hackers for the questions, and thank you, Professor Niema, for the talk. This concludes our tech talk today. Hackers, please come out to our MLH mini events right after this at 6 p.m. on Discord.